So hi everybody. So I will show you the work I've performed on two-photon scanless voltage imaging during my PhD. So as Valentina said, for all optical neurophysiology, it's important to be able to record the activity, either with calcium imaging or voltage imaging, but also to manipulate the activity. And so to image, we are mainly using fluorescent indicators. And so these indicators have the ability to change the fluorescence property to report the neuronal activity. And the vast majority of them are intensity-based, which means that the fluorescence intensity will change when there's some activity. And so you can report this change of activity with the so this can be expressed with delta F over F, which is the ratio of the change of the fluorescence to the baseline fluorescence. And so I will present really briefly calcium imaging and also voltage imaging. So calcium indicators are indirect reporters of the neuronal activity. As Valentina was saying, they rely on this rise of intracellular calcium that arises when there's an action potential in a cell. So they're very bright. They are located in the cytoplasm. And so when they bind to calcium, their level of fluorescence increases. So the latest variants are even able to detect a single action potential. But as you can see here, the kinetics of the calcium transients are much slower than the ones from the action potential. And so as a result, they're not really able to follow precisely trains of action potentials, and especially fast trains. And actually, when you have a look at the slowest variants, they're not even able to follow trains of action potentials faster than 5 Hertz. And so for this, if you want to be able to record fast trains of action potentials or also some subthreshold activity, you need to be able to, basically use voltage indicators. So these indicators are actually direct reporters of the activity. So they rely on this voltage sensing domain that it has to be located in the plasma membrane of the neuron. So they're coupled with a fluorescent protein, and when there's a depolarization in the neuron, there will be a change of conformation in this voltage sensing domain that will induce a further conformation of the fluorescent protein. And so this will lead to a variation of the intensity. So these voltage indicators are much faster. As you can see, the kinetics are on the order of the millisecond. So they are able to report more precisely action potentials, but also trains of action potentials. But they have some challenges that are linked to them. It's mainly that first, because they have to be located in the plasma membrane to be efficient, there's not so many molecules that can be inserted into a neuron. And so obviously you will get less photons from them, but also because we need to record faster to be able to image this fast activity, we get basically less photons. So as a result, most of the voltage imaging experiments that have been performed in the literature have been using single photon excitation and also wide field illumination, where the sample is illuminated with a beam that is not focalized. And because we're using single photon excitation, you end up with a lot of background fluorescence. So another approach has been to use targeted illumination, still with single photon excitation. And this has allowed to reduce the background fluorescence. But because the cone of illumination is still excited, it has basically been limited in depth to really sparsely labeled samples. And so another approach that had been developed is two-photon voltage imaging. So for voltage imaging, conventional scanning is too slow to be able to record the activity. And so different techniques have been developed. that can be divided into two main categories. The first one is random access microscopy, where a single beam is really rapidly displaced across pre-selected locations. And the other imply some temporal and or spatial multiplexing of the beams. So because these techniques are all based on scanning, and because we really work on a low photon budget, they all have been using PMT detections because they can track exactly the space where the photons are from where the photons are emitted. And so our project here was actually to use this two-photon scanless excitation approach and coupled with camera detection to be able to increase our photon budget, because we would be able to increase the dwell time on each cell, but also to record from multiple cells in the field of view. And so the work that I will show here is now under revision. And I have to mention that it has been done in close collaboration with the physicists from the lab. Okay. So, to be able to test first if this was possible, Ruth, so the physicist, has built a setup that can actually, for which we can use a GPC, computer-generated holography, or simple Gaussian beams. And here you can see that all of the different modalities that we tested ended up with the same profiles of excitation. And so the first question was to check if using scanless two-photon excitation was possible for voltage imaging. So we used cultured cells that were expressing this newest voltage indicator, GDR that was recently published. And so we patched cells and induced steps of depolarizations, either slow ones or fast ones, to mimic action potentials. And imaged with both of these three modalities. And so the results that we got, so first we could see that we were able to actually see these depolarizations with the imaging. So that was the first good news. And also what we could see was that for computer-generated holography, we managed to get a higher SNR for a given power. But also, we ended up with more variability because of the speckles. And so from now on, everything that I will talk about will be either done with GPC or computer-generated holography. So the next step was to check if we could detect real action potentials and what were the necessary parameters to image this. So we used organotypic hippocampal slices that were expressing the same indicator. And we induced action potentials that we recorded at 500 Hertz, 750, or 1 kilohertz. And we imaged at different powers. What we could see is that at every acquisition rate, we were able to distinguish with single trial action potentials. We realized that at 500 Hertz, we had a higher signal to noise ratio for a given power than at the others, which actually is good news for us, because this means that for imaging with the camera, we can have access to a bigger field of view. And also we can reduce the powers. And so in the case where we want to do multiple cells, we can have more power and image more cells. Okay, so this was for single action potentials, but as Valentina was saying, neurons can also fire fast trains of action potentials. And so we wanted to check if at 500 Hertz, we were also able to detect fast trains of action potentials. So we induced these trains of action potentials from 25 Hertz to 125 and recorded with the same parameters. And what we could see was that even at 500 Hertz, we had more than 96% of probability of detecting these action potentials. And also we had sub-millisecond estimation precision of the timing of the action potentials. One of the main advantages of voltage imaging is not only that you can detect action potentials, that you can also do with with calcium imaging, but it's the ability to detect the subthreshold activity. So it's these small events, small amplitude events that that neurons use to communicate between each other. And to see if we had any chance to detect some such small amplitude events, we patched cells and depolarized them. So performed depolarizing steps from 0.5 to 2.5 millivolts. And we did several repeats of this depolarization. And what we saw was that with only 5 repeats, averaging only 5 repeats, we were not able to distinguish the signal from the noise. But that after averaging several times, we could, in our condition, detect steps of subthreshold activity down to 1 millivolt. So these results, specific results, are to be put into context, is that the voltage indicator that we are using is not precisely designed to see to detect these small events, but actually there are some indicators that are designed to detect these subthreshold events. And so by using these indicators, we could actually maybe have better results. Then what we wanted also to check was if we could record some spontaneous activity, see if we could record also for a longer time. And so we patched cells and recorded the spontaneous activity here for 30 seconds. And we were able to detect single action potentials, trains of action potentials as here, but also hyperpolarizing events. Okay, so this was the part for detecting the neuronal activity. But as Valentina was saying in our talk, one of the main goals also of optogenetics is to perform optical connectivity experiments where you can photostimulate a neuron and actually record the response in a postsynaptic. So this works, this has been done, but one of the bias that you can get is that you can never be 100% sure that your presynaptic cell has fired an action potential when you photostimulated them. And so the idea here was to actually add the voltage indicator to the cell, to the presynaptic cell, and perform photostimulation and reading of the neuronal activity of this cell. So to do this, we use the same samples expressing Jedi, but we added the chrome, which is a rhodopsin, an excitatory rhodopsin, and we patched these co-expressing cells. And so by using the same laser beam that we were using for imaging, we were also able to activate the chrome in these neurons. And the results are here. So here's the electrophysiology trace. And we can see that when we turn on the laser, which is the pink bar, we were able to trigger action potentials, but also to image them. And here, as you can see, there's this small decrease in the level of fluorescence that here is zoomed out. And so we showed that we were able to photostimulate, but also record the action potentials. Okay, so all of these have been done in single cells using a high repetition rate laser source. But with the powers that we have after the objective, with these kinds of laser, we would only be able to record from 2 to 10 cells simultaneously. And so the idea would be to use low repetition rate laser that, because they have high peak energy, would allow us to reduce the average power and be able to image more cells. But most of the sources that are available commercially are fixed at 1030 nanometers. And this indicator has a peak at 940 nanometer. But as we can see here, on the action spectrum of the indicator at 1030, it looks like there is still a third of the action of the Jedi. And so we wanted to check if at this wavelength we would also be able to record activity. So we tried at 1030 with the high repetition rate laser source. And as we did before, we induced action potentials, and we could see that we were able to record in single trials this activity. So we used, so we then went to use a low repetition rate laser source at 1030. This is actually 500 kilohertz repetition rate. And we used computer-generated holography to image here 15 cells and to image the spontaneous activity in these slices at 500 hertz. And here you have the fluorescence trace of all of these 15 cells. You can see that most of them are firing synchronously, which is something that organotypic slices do. But you can also see that some events in some cells are not reproduced by others. And so we can see that this is actually not crosstalking between the mixed signals. Okay, so as a conclusion, we showed that two-photon scanless voltage imaging enabled the detection of single action potential with single trial, but also high frequency trains with sub-millisecond precision. We were also able to detect sub-threshold events, to photoactivate and record action potential simultaneously, and also to record multiple cells from multiple cells in vitro. And so this work can actually be super useful for the study of the connectivity. And with this, I would say thank you to everyone involved in the project. So mainly Ruth, who has worked with me on this, Valentina, and all of the rest of the team. Thank you.